everyone. Thank you so much for taking, an hour out of your evening, assuming you're in the West Coast here or in the East Coast here. Thank you for joining us. My name is Maggie Cameron, and I'm the Director of Development and Communications for the EHE Foundation. Before we get started, I need to share a few housekeeping items. First of all, this session is being recorded, so if you find you need to leave before it's over, that's no problem. We will post the recording on our website and YouTube channels in the coming weeks. Shouldn't take us too long. I also want to call your attention to the Zoom toolbar at the bottom of your screen. The most important down there is going to be the Q&A function. Please put any questions for our presenter in there. That's where we're going to be looking to pull questions for him. So you can do that as you go, as the presentation goes. If you hear something that you have a question about, please go ahead and put it in there. We are going to also leave time at the end for Q&A. So if we don't get your question right when you ask it, don't worry, we are going to keep an eye on that. And we have plenty of time at the end for Q&A. We also want to remind everyone that the EHE Foundation does not provide medical advice. The information provided here is for your information only, and we encourage you to contact your own healthcare provider for any specific questions about your health and participation in any clinical trials. Okay, so I think we're ready. Denise Robinson is going to get us started. She is the EHE Foundation's Director of Research and Interim Executive Director. So Denise, come on up.
Hi, everybody. Thank you, Maggie, and welcome, everyone. Thanks for joining this session this afternoon, this evening, wherever you are in the world. We are glad that you're joining us for this first session of EHE 360 Connect. Particularly with the growing landscape of clinical trials for EHE. We had originally planned this session to be included in our annual conference in April and had to make a last-minute change. So we're delighted to have Dr. Greg Cote here today, particularly as the landscape of clinical trials for EHE continues to grow. Many of you may be familiar with Dr. Cote. He is a clinician scientist and medical oncologist at Mass General Hospital Cancer Center in Boston. He is an active translational researcher and early phase clinical trialist, and is a trusted advisor to the EHE Foundation. Importantly for all of you, Dr. Cote is highly experienced in the treatment of EHE and other sarcoma subtypes. As Maggie mentioned, I want to encourage you to put your questions in the Q&A along the timeline while Dr. Cote is talking. He may stop and pause and pick any of those up, but then we'll have plenty of time afterwards to go through those questions and talk with you. So I'm going to turn it over to Dr. Cote now. Welcome. Thank you so much for being here tonight.
All right. Thanks so much for the invitation and opportunity to speak to everyone today. Really great to be here. So let me just get my slides rolling and then we'll jump right into it. So I've been tasked today to discuss with you about clinical trials, and this is a very complicated topic. So I'm going to give you a high-level view. We'll jump into some details, but as you heard, feel free to fire away some questions. I'll try to keep an eye on the Q&A area and pick some up as we go, and if not, we'll catch up at the end. So let's get this going there. Here we go. So just to set the stage, many of you already know this, but the sarcomas are rare. These represent about 1% of all cancers. That's somewhere around 12 to 16,000 cases per year. And what's really mind-boggling is that within that 1%, there's 70, maybe 80 different subtypes, including EHE. And all of these have distinct biology and different behavior and different medical management. And when you think about that from a drug development standpoint, this is really challenging, right? We have lots of different subtypes. They're rare. They behave differently, very different than what we see in something like breast cancer or lung cancer, where there's hundreds of thousands of patients per year. And you can run large trials and really figure out, what's the best way to treat these. We're looking at a fraction of patients, maybe, I don't know, a few dozen per year that need systemic therapy. So a lot harder for us to run these clinical trials. There are other challenges as well. So we're missing some of the targets that we see in other common cancers. What do I mean by this? So it turns out that different carcinomas, be it breast cancer, lung cancer, some GI cancers, they have mutations in these signaling cascades, these signaling pathways that are common among them. And if a drug company spends a lot of money and develops a drug that targets this MAP kinase pathway in, say, GI cancers, well, it turns out there's also these mutations in lung cancers, so they can, research that drug in those as well. We're really missing that in EHE. We don't have those same mutations, although this is changing. And this is particularly important when we talk about TEAD inhibitors, and we'll get into that a little later. Typically, small diseases like EHE are a lower priority for drug companies. It's not difficult to understand why that might be. These are rare, so it's a small market, unfortunately. So they're not going to recoup their investment, but they're also challenging to treat because we understand to develop drugs, and because we don't understand the biology as well. So these are challenges, but with any challenges, opportunities, and the sarcoma medical oncology community and patient community are very interested and dedicated into developing new and effective and tolerable therapies. And we've made quite a bit of progress, and even in my short career, and I'm hopeful that we're going to find some new treatments soon that will change the landscape for this disease. So let's jump from there into clinical trials. So just broadly, what is a clinical trial? So this is where we're trying to take a treatment, some drug. It could be a brand new drug, it's never been in people before, or maybe a drug that's approved for another type of cancer that we want to look at in a particular disease. And we're trying to match that with these different tumors to develop new treatments. So this could be a new type of chemotherapy matching with a patient population, could be a new target that's hitting some unique part of the biology of a cancer. And when we try to run clinical trials, we're trying to develop these to show that they work and eventually get these FDA approved so that any oncologist can prescribe them. All right, so let's jump into more detail here. So this cartoon is a schema of sort of the life cycle of a drug. And this is a complicated process. It can take 10 years, billions of dollars, but it's sort of the best we got at this point. And hopefully you can see my pointer. So things start in the lab here in the preclinical research area. What do I mean by this? So this is where a scientist in the lab may be treating different models of cancer in petri dishes. We call them cell lines, or maybe mouse models of different cancers. And what they're really trying to do here is show that they can hit this target and get these tumors to shrink. If that looks good and looks encouraging, it looks like it's a real drug, a chemist might make some modifications. And then eventually they do these toxicology experiments where they try to see if there's any unexpected side effects in different animals. And then eventually, whoever is developing that drug, could be big pharma, a small biotech, could be an academic institution, or even an investigator, would bring that package to the FDA and say, hey, I want to develop this drug in cancer patients, and they go through this process to start a clinical trial. So the different steps of a clinical trial are called different phases: phase 1, phase 2, phase 3, and they have different goals at these points. And we'll go into a lot more detail on this in a minute. But in general, in phase 1, the question we're trying to answer here is this drug safe, and what's the best dose? We have no idea, right? This drug's never been in people. It may look great in a mouse, but we have no idea if it's going to be safe or it's going to work in people. So we do these very careful studies, and I'll give you some examples of that soon, trying to figure out these questions. At the same time, though, we're really also hoping to see if it works, because why develop a drug if it's not going to treat your cancer? So as we go through, in addition to trying to answer these questions about safety and dosing, we're trying to see signs of efficacy. As this drug matures, once we find the right dose, we do something called phase 2, and this is where typically the same type of cancer patient or same mutation will get that dose that we figured out in phase 1. One bit of nuance here. So in phase 1, typically this could be any type of cancer. They mix and match, or maybe a limited subset, or if there's a particular mutation that a drug's going after, they may say, okay, any type of cancer can go after this as long as they have that mutation. In phase 2, this is typically narrowed down, where we may say the drug's only going to be used in one type of cancer: pancreas cancer, lung cancer, something like that. Or occasionally it might say they have to have all exactly the same mutation. Most of the time in all the time in phase 1, and most of the time in phase 2, there's no placebo. Everybody's getting the drug. So once we see some level of activity in phase 2, things are looking good. That's when the drug moves to phase 3. And these are the large randomized trials that you might see in the news. So this is where we're comparing a drug versus placebo or versus some standard of care to prove that it's actually better. And if that looks good, it goes on to FDA approval. So quite a process. It takes a long time, but it's the best we have so far. Now, there are some variations on this, and sometimes drugs can get approved without phase 3 studies, but we can circle back to that in the Q&A time if people are interested. All right, coming back to phase 1. So this is, again, where we're trying to figure out safety and dosing. What's the right dose? Is this drug safe? What are the side effects? And hopefully we see that sign of activity. So there's lots of different ways to run phase 1 studies. The most classic is something called 3+3. I won't bore you too much with the details, but let me show you some cartoon models of how we think of these. So in this example, the circles are individual participants in the study, patients with any type of cancer who join. If the circle is higher up, it's a higher dose, and then time is as time goes along. So we pick a starting dose, and this is based off of some predicted dosing based on animals, and it's a very low dose, and we treat just three people across the world, and we see how they do. Usually there's a defined window, typically something like 4 weeks, and if they do okay, then we increase the dose and treat 3 more patients. And they do okay, we increase the dose and treat 3 more, and so on. Eventually, you get to a dose level where people start having side effects, and if it's a serious side effect, it stops the dose escalation, and we say, okay, wait, something's going on here. Maybe this dose is too high. Now, it's only 3 patients, so it's hard to make a lot of conclusions here. So in this type of dose escalation model, we would accrue 3 more patients at that level, see how they do. And if they do okay, then we can keep going. And eventually, you'll reach a dose level where too many patients are having side effects. In this example, it's 1/3 of the patients. And you say, okay, we got to stop here. This isn't safe. And at that point, the company, the investigators, the people working behind the scenes will get together, look at all the data, all the drug levels in people's blood, side effects, tolerability, efficacy, how's it doing, working to treat cancers, and we'll declare what we call the phase 2 dose, the dose we want to take to that next step. And then from here, it keeps going. Most studies will have what's called a dose expansion, where we treat more patients at that level to confirm the safety and sometimes try to see more of a signal of activity. These tend to be very busy studies, as you might imagine. So it's only 3 patients at a time, but we need to make sure they're safe. So there's a lot of extra blood draws for safety. We check EKGs. We're also measuring the level of the drug in somebody's blood. So if you take it by pill or by IV, we have to know how that's absorbed and cleared. So very complicated studies. Sometimes there's extra biopsies that are included to see if we're hitting that target that we think we're hitting. But eventually, using these designs, we'll figure out more, we'll understand more about this drug, and then we'll know how to move forward. All right, I see one question in there. Is the FDA solely for the US? Is it the same process in other countries? Yeah, so it depends on the country. So the FDA is for the US, the EMA is for Europe. Other countries have their own regulatory bodies that approve drugs. Some will accept guidance from the FDA. Some will accept guidance from the FDA, EMA. Some will need to have their own studies run. But every country is different, correct? All right, so coming back to phase 1, we have this conflict where we're always trying to balance the potential toxicity of a drug versus treating somebody at inactive doses. So what do I mean here? So see if I can go backwards. Hang on. There we go. So if somebody's treated at the first couple of doses, you might say, all right, well, this is probably going to be safe because it's a very low dose, but maybe a lower chance that it's going to hit the target that we're looking for. Whereas if somebody's treated at the higher doses, higher chance they might have a side effect, maybe higher chance we hit the target. And we don't know beforehand like what the sweet spot is. So, as investigators, as people run these trials, we have to accept this. I will say that I've had plenty of patients who've been dosed in early parts of studies where if it's the right drug, the right mutation, the right patient, where we see activity. So, it's still definitely worthwhile for the right situation to participate in studies early on. And then the second part is that it depends on the study, but some studies will allow patients who've been at lower doses and are doing well to jump up to higher doses as well. All right, so now let's talk about phase 2. So as I mentioned earlier, the dose here is determined from phase 1. Most often, this is everyone with the same type of cancer, could be the same mutation. And here we're really looking to build on that signal of activity that I mentioned to support a phase 3 study. Typically, these are not randomized studies. Usually it's just one arm, and we say, okay, we need to see this many patients do well to be confident that this is a real drug and worth pursuing further. So the best example of this in EHE is the trametinib study that was published earlier this year. Perhaps some of you participated in it. Trametinib is a drug that's approved for the treatment of cancers like melanoma, skin cancer. And based on some laboratory work, it was predicted that this drug may actually be helpful for patients with EHE. The investigators for this study plan to accrue around 40 patients. They would treat them with imatinib, no placebo, everybody got the drug. And then after a period of time, they would get what we call a response assessment. That's just getting CT scans and MRIs. Now, studies have different criteria on how to measure that response. Most of them use the same response criteria that we can go into if people are interested. But basically, you're trying to say, is the tumor shrinking, or is it stable, or is it growing, or something new showing up? And for this study, the investigators said that, okay, if 20% of these 40 patients have tumor shrinkage, hey, this is a real signal. This is better than anything we've seen in EHE. We should develop this further. It would be difficult to get something FDA approved off of this small cohort, but it could enter what we call our guidelines, where if a drug's approved for other cancers, we can prescribe it off-label, and it will be covered by insurance companies. If it's less than 20%, okay, well, we're not hitting the bar that we're hoping for, and we would stop development at this point. So unfortunately for trametinib, did not meet the mark and development stopped, but it's a good example of phase 2 study for EHE. All right, I don't see any questions, so I'll keep rolling. So coming back to this schema here, as I mentioned before, this is quite a process. 10 years, billions of dollars. So there's a lot of pressure in investigators and industry to make what we call like a go, no-go decision. What we don't want to do is get to phase 3, spend $1 billion and have a drug fail. Like, we're really not helping the community. We're wasting everyone's time. We're wasting money. We're wasting patients' time. So what can we do to help that? So there's a lot of pressure now trying to build up the efforts we have in phase 1 and early phase 2 studies to try to move this decision-making along early. And so what that might mean is that built into these phase 1 studies, once we think we have a phase 1 dose, we actually will run multiple phase 2 studies as part of this. So studies that may have been like 30 patients 15 years ago can be like 150 patients now. So huge studies. And in this example that I just made up, we have a drug, we figured out the dose, and we're going to run multiple phase 2 studies. One for breast cancer patients, one that have a mutation in a gene I made up, and then hopefully another cohort that has EHE patients. And then the idea here is that these phase 2 studies will tell us, okay, we're seeing enough activity here. It's time to move this to a larger study to push the development forward. All right, confusing stuff, but we'll keep going. So let's come back to EHE patients. So what paths might you have to participate in a clinical trial? So we hope someday that we will have enough patients and enough knowledge and enough active drugs to run phase 3 studies, but we're really not there quite yet. There are some concepts that may get there, but right now, really the focus for patients who want to participate trials is in early phase studies, so phase 1 and phase 2. So this could be a phase 2 study where an investigator says, okay, there's this drug, it's again approved for a different kind of cancer, and I want to see if this works in EHE. They may write a study and you that, or it could be one of these phase 1 first in human studies that may be important for EHE. And you may say, okay, well, why would I want to participate in that? It's a brand new drug. Well, if the science is good enough, like I said, or the drug is exciting enough, maybe some EHE patients have already been on there, then it's going to be worth your time. So how do you find these studies? And this can be pretty dizzying for patients, and you need to lean on your EHE organization community here, or on your own doc. But you can speak to your oncologist if they're an academic institution. If they're at an academic institution, they may be running some of these studies. If they're not personally involved in the studies, they may be able to refer you to somebody within their institution. The fightehe.org website has a link for clinical trials actively looking for EHE patients, so you could go there. The NIH, the U.S. National Institute of Health, runs a website called clinicaltrials.gov. This is a little clunky, although it's getting better. But every trial that runs in the US must be registered there. So you can actually search in there and type in EHE, filter for recruiting, and you may see some studies that are looking for this. Sometimes, though, there could be studies that are important for EHE patients that may not even have EHE listed. So that's when you're going to need to lean on your oncologist or the EHE organization here. The last thing I was going to say is that if you're being treated in the community or even at an academic institution that doesn't have trials open, it's still worth asking your oncologist, because they may be able to refer you to somewhere else to go on trial if you're able to travel. All right, so I wanted to spend a couple minutes and talk about the HIPPO pathway, because this is probably one of the more exciting areas in drug development for EHE right now. So taking a step back and just thinking about the biology of cells, we actually, within your cells, normal or cancer cells, there's these we call signaling cascades. So these are, it's almost like a game of telephone, but like one protein talks to another protein, talks to another, talks to another. And eventually that triggers hits the DNA and turns on a gene, or turns on a DNA gene program and modifies that cell. And these have so many different steps because they're so tightly regulated, because if something goes wrong in these, it could lead to something bad happening, like a cancer growing. And we see this across cells, across cancers, where typically something's been mutated and that leads to the cancer growing. And so for this pathway, this is conserved across many different species. And this starts with this protein called NF2. It signals down through LATS1-2, YAP-TAZ, and then YAP-TAZ binds something called a transcription factor called TEAD. And TEAD actually binds to DNA and turns on different programs. And interestingly, we see cancers where there's mutations at all stages of the way. For NF2, for example, that's a sort of a brake pedal on the system. So if that's mutated, it takes the brakes off, and then there's uncontrolled signaling down this cascade. Or in the case of EHE, there's these gene fusions, which I'm sure you're aware about, where two genes get stuck together that shouldn't be together. They have funny names like TAZ, CAMPTA1, or YAP, TFE3. These actually enter this pathway, and it's almost like the gas pedal's stuck on, where they keep binding TEAD and signaling. So some clever chemists said, can we block this interaction? And the answer is yes. There's many different drug companies looking at this right now and trying to develop drugs for patients that can interfere with this YAP-TAZ binding of TEAD and thus shut down the pathway. Now, these companies are mostly interested in big diseases. So NF2, it turns out, is mutated in a good percentage of mesothelioma you may have heard of. So that's the cancer that's associated with asbestos exposure. And it's also activated in some tumors that have different mutations, but use this as a resistance pathway. But that's okay. This is great for us, because if big pharma or biotech is interested in this pathway, we can latch onto it and try to develop drugs for EHE in parallel. And we can do that by putting patients on phase 1 trials of these drugs. We can do that by, once they have an actual dose, trying to build out a cohort of EHE patients, and we're doing that now. So as I mentioned, multiple drugs are being developed in this space, and there's different companies and some of these are listed on the EHE website. Most of these are in early phase, so we may not know the right dose, we may not know the right side effects, but I think if you're looking for a trial, this should be a high priority of things to think about. All right, we have a question. So in order for a patient to participate in a clinical trial in the US, should I be a US citizen? Yeah, this is a tricky one. So it depends on the country you're in, but in the US, the way clinical trials work is Anything that's considered standard of care, so routine labs, routine scans, routine visits with me, that's covered typically by insurance. And then anything that's considered research, the study drug, research blood samples, research biopsies, things like that, that's covered by the trial. So this model is different in different countries, but that's the way it is in the US. So if you do not have international insurance, this would be cash billed to you, and that the numbers can be extraordinarily high. Generally that's not feasible, but it may be something where one of these studies may open up in a country where you live, or in another country where there is healthcare studies covered by healthcare by the government or something like that. All right, so next question was about should oh, it disappeared. Participation in EHE study would only make sense for patients with clinical manifestations of cancers. Okay, yes. So let me touch on that in a minute. So basically what's being asked is if your cancer is growing slowly, should you participate in a trial? We'll come back to that, okay? All right. So say you're interested in a trial and you decide to sign on. Your doc will talk to you about something called informed consent. What does this involve? So you actually will get a document. It's written in plain lay language, although there's some medical terminology, no way to get around that. But basically, it's a long document, 20, 30 pages that says, describes what we're trying to do. It's all about the drug, what the reason we're doing this trial, what's expected of us as the clinicians, what's expected of you as the patients. It'll give you the schedule, things like that. When are the scans, when are the biopsies, what are the expected side effects if they're known? And you'll have time to go through that, time to ask questions. And if you want to move forward, you'll sign the dotted line. If you say, hey, thanks, but no thanks, this looks a little too experimental for me, I'll do something else. That's okay. Like, your doctor will still be your doctor. They're not going to stop being your doctor because you said no. Just have an open conversation with them. If you're feeling pressured, maybe the answer should be no. So take your time and make sure you understand what you're signing up for, because we want you to be an active participant in this. If things go forward and you sign on the dotted line, then the next step is called screening. So what's screening? So every trial has a set of eligibility criteria and ineligibility criteria. These are things like checking routine lab tests, EKGs. Do you have any other medical problems? Are you physically fit, or are you critically ill, things like this. And the reason we have to go through this is we want to make sure that it's safe for you to participate in the trial. So say, for example, we have a new drug and we don't know all the side effects and there's some bad effect on, say, the kidneys. Well, if your kidney function is normal, maybe you can tolerate that insult and recover. But if you already are starting with bad kidney function and it gets worse, you may not recover. So we got to put these criteria in to try to be as safe as we reasonably can. So we'll go through this list. And most of the time before somebody signs on the dotted line, we've done a chart review, so we already have an idea if somebody would be eligible. And if things look good, then we go for the treatment. Now, this treatment could be very different depending on what drug it is. Some are IV therapies, some are pills twice a day, some are pills once a day, and so on. So the schedule will really depend on the drug and the trial. Typically, you'll get a study calendar. They'll tell you when you need to be there. Like I said, what's expected of you. We generally are more involved in the day-to-day checking in of our patients who are on trials, particularly in phase 1, just because these are new drugs and we don't know all the side effects. So there's a lot more check-ins than you might be used to if you were on standard of care. All right, let me just take a look at the Q&A here. So one question is, are there specific drugs that a prospective participant should avoid to ensure that they're eligible for a trial? Yeah, that's a good question. So that depends on the study. For most phase 1 studies, they may just say not in the same class of drugs. So if you are already on a TEAD inhibitor, most other TEAD inhibitor studies would not allow you to participate again, although you could argue that maybe doesn't make sense, but that's the way it is. If there's a future mTOR study, which there may be in the future, they may restrict patients who've been on prior mTOR inhibitors. But I think with what we know now about EHE, I think, whatever you and your oncologist decide is the best treatment, you should go forward with. And I wouldn't modify things yet, just because we don't know the answer. Down the road, we may say, okay, wow, this drug is way better than that drug. So, don't go on these other drugs. So we don't want to make you ineligible for this trial. It's clearly better. But right now, with the information we have, we would hold off on that. Yeah, that question was asked twice. So, one question was like, are there any trials coming up that may not cause neuropathy? Yes, I would say the answer is yes. A lot of the drugs that are in development do not typically cause neuropathy. If it's predicted that there might be neuropathy, then those trials may not be the right fit for you. All right, I'll keep moving. So key points here, and I can't emphasize this enough, is how important this informed consent process is. So patients may say, "Oh, am I a guinea pig by taking part in this?" And the answer is no. Like, you are a participant. Like, this is your decision. Obviously we're coming at it from different angles because we're on the doctor side and you're on the patient side, but ultimately this is your decision, and we'll give you our thoughts, but ultimately you get to decide if you want to move forward or not. As I said earlier, most patients for EHE are participating in phase 1 and phase 2 studies. So as things are right now, it would be unlikely that there would be any placebo involved. You could envision a time where we could have some small set of patients that are randomized to placebos for a trial. But this would really only be to get at the sort of the heterogeneity of EHE. What do I mean by this? So some patients with EHE will have their tumors grow very slowly over a long period of time. Others will go quickly. And so there might be a need, depending on how active a drug is, to have a study design where there is a placebo. But as it sits right now, that's unlikely to happen. And then again, these early phase visits, really exciting technology, really cool drugs, but there's a lot of unknowns here. First time these drugs are being given to people, they tend to be very busy, lots of visits, lots of blood draws. Sometimes those require biopsies. And then these later phase studies are less busy, more because we have more prior experience with the drug, and we know more about the side effects. So it's a little less is asked of you. All right, so I wanted to come back to how I think about a new patient with EHE and how they might, where trials might fit in. And this gets to Alex's question here. So when I see a new patient, really important, I want to confirm the pathology. Now, there's a lot of amazing pathologists around the world, but EHE is a very rare diagnosis. And I've had cases where people have been diagnosed with one thing and then came to our institution and they changed the diagnosis, sometimes vice versa. And so it's really important to carefully look at the pathology. And then with modern technologies, we can look for those gene fusions I was mentioning and prove that this is EHE, because we want to know what we're treating. If you're being treated in the community, that's totally fine. I know a lot of community oncologists who are amazing clinicians, see incredible breadth of cancer and take great care of their patients, but they may have never seen EHE before. So in those scenarios, like what I'll do is I recommend they see a sarcoma expert at least once, maybe twice, and that person can be involved with the community oncologist so that they can, ask questions back and forth. So if I have a patient who lives, 6 hours away, they come down to see me, I give my thoughts. If they're not going to go on a trial, they can go back to that oncologist and I'll be involved to help make decisions. And then this is probably less important for EHE than it is for other cancer, other sarcomas, but I'll often consider genetic testing where we're looking at the genes that in that tumor to see which are mutated. Most EHE are driven by those gene fusions, so the other mutations may not be as important, but it's still reasonable to think about. Okay, so then we get to this question, Alex's question here. So observation versus other treatments. So as I mentioned before, some patients can have metastatic EHE that just sits there, and I have patients who it hasn't been growing for years, or maybe it's like a few millimeters at a time, where they may not need treatment for a really long time. Other patients can kind of grow slowly, and other patients can take off quickly. And so for us, the sort of sweet spot to start therapy is when things start growing, but before they start growing too rapidly, because we want to sort of cut that off before things get out of control. So we may decide to do an observation strategy, and that might be just getting imaging in 3 months. And then if that looks good, 3 months later, and eventually start spacing it out to 4 months, maybe 6 months, and so on. There's some patients we may think about surgery. You probably know many patients will present with metastases, but if there is an isolated lesion, we may think about surgery. Occasionally, we'll do radiation to any lesions that may be symptomatic. And at some point, somebody needs to think about systemic therapy. And for me, and there's different strategies with different oncologists, I always ask the question first, is there a clinical trial? Because in my mind, we're not quite as good as we'd like to be as far as the available sort of off-label therapies. And if there's no trial, then we'll do some systemic therapy of either chemotherapy or mTOR inhibitors or some other targeted therapy. And we'll do that for some period of time. And if at some point we'll need to move on to something else, if there's a trial available, then we're going to look for, is this a phase 1 trial that we can jump on that makes sense for EHE, or is there happen to be an EHE trial that's open, like the trametinib study, and we'll jump on that. And eventually that may stop working. You kind of go to the next, to the next, to the next. And the hope is that you can string together, one of two things happens. One is that you string together all these different therapies. So we observe, we do a trial, we do chemo, and this is a long period of time of keeping things under control. Or we go through this roadmap here, and maybe one of these trials really hits and really change the landscape of your disease. And so that's the hope for the future. So why don't I stop there, and I can take other questions from the group. I might have missed some that along the way.
Thank you. That was great. That was great. Alex's question regarding clinical manifestations and whether the disease is in stealth mode. Kelly has just asked a question regarding, can a patient access a trial from a hospital other than a hospital where the primary researcher physician
Oh
is?
yeah, so that's a great question. Yeah, I probably should have touched on that. So yeah, so some trial, it depends on the trial. That's the long story. So some trials may be at like one institution. So say we had this trial of this drug called Eribulin. It's a chemotherapy drug approved for breast cancer. That was a trial that I wrote, and so that was just at my institution and at Mass General, and then one of our sister institutions, Dana-Farber. And so if you wanted to participate in the trial, you had to come see us. However, the trametinib study was run through a cooperative group we have called SARC, and they opened I don't know, Denise, do you know, is it like 20 sites or something like that? Yeah, across the country, across the world. And so it really just depends on the study. So a study could be my idea, and I try to run it here, or maybe at a couple institutions because I can't run it across the world because I don't have the funding, or it could be from a drug company, and maybe they want to open 30 sites. And so what they'll do is they'll start looking around for people who see a disease of interest. So if we use EHE as the example, they'll ask the different institutions around the country, around the world that take care of EHE patients and say, hey, we have this idea, do you want to participate in it? And, we'll either say, yes, that's interesting, or yes, it's interesting, we don't have the bandwidth to do it, or no, we'll pass. And so that's any particular study of interest. You can only participate where it's open. So if some investigator opens it up in Colorado and in Boston, and you live closer to Colorado, go there. If it's only open in Boston, then we have to figure out the travel logistics. Some studies, particularly those that are run from industry, will support people for travel. And I think this is extremely important for ultra rare diseases like this. And so when I do a lot of phase 1, if you didn't gather, and when I talk to companies, I always ask them to include a travel budget, because it's a big ask for people to be in the hospital so much. So it's helpful if they can support that, particularly if people are traveling from a distance.
Great, thank you. Tanya asks, did you mention any current EHE specific trials? Are there any currently?
Right, yeah. So I don't think there's any specific that are only looking at EHE, but some of these TEAD inhibitor studies will take EHE patients because it's right on the way they're looking for. So they're looking for cancers that have mutations along that Hippo pathway. there are definitely some in design, and like we've had a lot of meetings. We were just talking about this just before the Zoom started. That there's a really cool concept one of our colleagues is working on. We're trying to figure out the funding and the number of sites, and so there will be some down the road for sure. But right now most of it's in an early phase. Denise, did I miss any? Do you know of any specific that are open?
No, I don't. And I was just going to say for Tanya and all, I think Maggie popped into the chat earlier. Go to the fightehe.org website, go to the research section, drop down to clinical trials. There are 4 or 5 trials listed there. One of those is a registry study that's taking place in Europe, but still important if you're in Europe to know where those locations are, because that is a contribution to advance, drug development and repurposed drugs too. So take a look at that. Fiona asked, when a trial goes into phase 2, can it become international? So this
Yeah.
one.
So again, it just depends on the trial and the funding. So if it's a small trial, individual investigator, typically these are not international just because it's very difficult to do that. So the way we got around that for that eribulin study I was mentioning, so a colleague of mine in Australia had a very similar idea as me. So we decided to write the trials exactly the same, where they ran theirs independently and ran mine independently. We're publishing it together. So, but it's very very difficult to do that for individual investigator. If it's a drug company sponsored study where they have exponentially more funding, then yes, they can definitely do that. So say envision a time, and I hope this happens. I don't know if it will, but envision a time where one of these TEAD inhibitor drugs is the winner and is interested in EHE. We will probably run an international trial to treat more patients and hopefully prove that this works.
Yeah, and several, I think at least one of the studies that's listed on our listing right now, the Novartis trial, is an international trial. That one is maybe 5 countries or so right now. So take a look at that one. Harriet asked, on your last chart, and I think she might be referring to maybe her something in her chart. What is the difference between chemo and targeted therapy?
Oh yeah. It was the slide
chart
I had about Okay, so good question. So we're in our from an oncology side, like our spare time, like all we do is think about ways to kill cancers, right? And so chemotherapy are drugs that for the most part are designed to bind to DNA and disrupt it in some way. So this could cause it to break or interfere with how those genes are turned on, do something. And typically cancer cells are more sensitive to that disruption than a normal cell. Not every normal cell, though, is resistant. So that's why people lose their hair, have low blood counts, nausea, things like that. That's where the side effects come from. Some chemotherapies will also bind these things called microtubules. These are like little kind of scaffolding proteins whose job it is to kind of hold the cell together. And that's really important for vascular sarcomas like EHE and angiosarcoma. And basically these drugs will bind those and disrupt their function. And then there's some chemotherapies that target some metabolism pathways as well. But generally these are toxins. I mean, they're poisons. We're trying to poison the cancer, but that's how they work. But targeted therapy is a little different. So these are drugs that are designed to bind a particular protein in the cell. And usually these are, in the perfect situation, a mutated protein. So something's gone wrong, the cell's stuck on, and you have a drug that kind of combine that to stop it from working. So, we don't have the perfect targeted therapy yet for EHE, and that's what we're trying to do with these TEAD inhibitors. But for other cancers, this is extremely important. So for some vascular tumors like EHE, we will use these, we call these dirty tyrosine kinase inhibitors. Why they call it dirty? Because they bind a lot of these different proteins. And the hope is that we can find one that binds proteins that are important for EHE cells. So an example of that would be a drug called like Votrient or pazopanib you may have heard of. Hopefully that answers the question.
Okay. That was great. Thank you.
All right.
Hopefully everybody got that. That's a lot.
It's a lot. Yes,
big
exactly.
Big science session. Christine asked, what is the Hippo pathway? Wow.
Yeah.
How much
All
time
right.
do
Let
we
me go
have?
back to that slide because that's confusing. So that was up here somewhere. There it is. All right. So this is the Hippo pathway. So what the heck is this? All right. So as I said before, and I'll go through this again at the risk of being repetitive, just because it is confusing. So in every cell in the body, cancer or not, there are these, we call them signaling cascades, and it's basically a way for in a certain situation for a cell to turn on a genetic program. So the way your biology works, so you have DNA, those are your genes, that's your genetic programming. And that gets turned into RNA, and that gets turned into protein. And protein are the parts of cells that kind of have function. And so in this example, the signaling cascade is a protein called NF2, signals to MST12, signals to LATs, signals to YAPTAZ. And YAPTAZ goes into the nucleus, that's where the DNA is, and interacts with this protein called TEAD, and that binds to DNA and turns on a program. So in a normal cell, that's a really important sort of growth signal pathway that tells a cell to grow and it shouldn't grow. So in some cancers, there's mutations along this pathway. And in EHE, the gene fusions that are seen in EHE are called TAZ and YAP, bound to different proteins. They displace normal YAP and TAZ, bind TEAD, and sort of turn that pathway on. And once it's on, it stays on, and the cell's growing because it's constantly getting this signal. Hopefully that makes sense. Why it's called Hippo. So this, I think it was from Drosophila. It's like the fruit fly originally. And so when researchers at the time, you turn that pathway on, the fruit flies got big, like a hippo.
Yeah, yeah. Okay, let's see. Thank you. That was great. Fiona asks, what about other sarcoma trials that may be open to EHE? She's mentions an immunotherapy trial opening at Princess Margaret. Fiona's in Canada, by the way. And she asked, would that be a basket trial?
Yeah. So this is a great question. So some studies may open up multiple, we call them different cohorts for different types of sarcomas, or allow different types. And absolutely, if there's an option like that that looks interesting, that is recruiting EHE patients, then by all means, that should be looked at. So the Eribulin trial, I keep going back to that. We haven't even published it yet, but in that study we allowed either angiosarcoma or EHE. So it's like a small basket trial, but same idea, right? So you could have any of those vascular types of sarcomas and participate. But yeah, there's other trials out there as well. In the old days, we used to, is before my time, but when a sarcoma trial would open, it would be typically chemotherapy based and allow any type of sarcoma. And as I said, you have 70 or 80 different types. So within those studies, you would get at least a dozen different types of sarcomas, and it would be very hard to interpret. So say you saw something interesting in a single EHE patient, well, we don't know, was that a one-off thing, or is that something real that needs to be chased? And so many of us, when we run trials now, we recognize that it doesn't make any sense. So we're parsing these out to individual diseases. And I think what's really cool is that, if you would ask the community 10 years ago whether a study EHE could be done, people would say no. But then the tremendous study was successful, right? I mean, there's a negative study, but they still prove that we can accrue these types of studies. And so I think that's been a really important outcome of these studies, even though the trials were negative.
Yeah. Really important to have a number of people to study that have like or similar presentations of the disease, so that we can learn something.
Exactly.
Even in those basket trials. That's really the key. Jennifer has a question, and before I wanted to pop in a question that someone had asked me offline. I want to touch on this. Can people who have had a liver transplant get into a clinical trial?
Yes. So again, it depends on the specific trial. So there could be some exclusion. So say it was an immunotherapy based trial, right? Those typically patients with so what is immunotherapy? So we didn't really talk about this. So this is another class of therapies, but the idea there is to take the brakes off the immune system, to let your own immune system attack EHE, or some are kind of stimulatory where they try to rev up your own immune system. So if you've had a liver transplant, you're getting somebody else's liver, so you take immune suppression so you don't reject that liver, so your own immune system doesn't attack it. So if you were on an immunotherapy or looking at an immunotherapy study, you might not be the best candidate because we don't want to have you A, have immunosuppression and not have it work, or B, reject the liver that you got. However, for other studies that are targeted type therapies or even some chemotherapies, as long as the liver function's good and there's no contraindication like drug-drug interaction with your immunosuppression, then it should be fine. And I've had patients with liver transplants take part in trials. We just need to be very mindful we don't hurt that liver, because that's, you know, obviously important.
Got it. Thank you. Thank you. Okay, Jennifer's question. Can you tell us about why the EHE study was discontinued, and had the medication been helpful for your patients? And I'm going to just say right there that I don't believe that Dr. Cote can say very much about this. You can add what you like, but I think
yeah. So I can't really say anything other than what's in the press release, but just thinking back about that decision to go or no go, companies are looking for certain metrics to see if they're predicted to be successful if they go forward. And while I can't talk about that specific example, just broadly, if a company feels like the drug's not as reaching the bar they want to hit, they may stop that program. And it's tricky because you know, there could be patients deriving benefit. There could be you know, a subset of patients we haven't studied yet where we think this drug could work, and it's very frustrating for investigators and for patients. Occasionally, there's situations where there's other drugs in that space, and you know, we'll jump on that. Or for another tumor we take care of called desmoid, the drug can be picked up by another company. So there's this drug called nirogacestat that was recently FDA approved for desmoid tumors that was discarded by Pfizer. They weren't going to move forward with it, and another company licensed that drug, and it led to FDA approval. So I can't get into specifics about EHE, but you know, these are for-profit companies. In the case of EHE, it's a small company, and so they have to be very careful about what they do. You know, even though it may be. Well, I'll just stop there.
Yep, that's good. That's good. Christine asks, I have a second cancer, CML, which is also a gene fusion. She's taken imatinib. Any connection between these two cancers? She says her EHE is stable, thank goodness, and not growing. So she has CML, EHE, and she takes imatinib.
Yeah. Okay. So this is a great question too. So let me take this from a couple different angles. So first off, from a trial standpoint, sometimes they will restrict patients who have two cancers, and particularly if you're on a therapy for that other cancer. So that could be an issue for trials. We're starting to see some trials put in more friendly language where if there's a diagnosis of a cancer, but it's not going to impact the study in any way. So say it's like a slow growing indolent skin cancer, or maybe somebody was treated for a breast cancer that was very early stage, then patients will still be allowed to participate in studies. For CML, if somebody's on therapy, that may be a little tricky, but some docs will, when somebody's in remission, hold the drug, in which case you could participate in a trial while that's held, as long as the right language was there. Could the CML and EHE be related? I think probably not. There's different biology in how these tumors grow. However, similar to both are mutations that happen that drive these cancers. So I suppose, you know, you could think about, like, could there be some predisposition to developing these mutations? I don't know. It's hard for me to answer that without knowing more about the situation. Could the imatinib be treating the EHE? Also an interesting question. So imatinib is a drug that binds to some very specific proteins. That's a targeted therapy, like I was talking about. It's not known if those particular mutations are important for EHE, but it's possible that another target that we don't know about is being blocked by that. I don't think imatinib's been studied in detail for EHE. When we give drugs like pazopanib, Votrient that I mentioned earlier, that hits the same targets as imatinib plus probably like 30 others. And so it's very possible there's some overlap. Or the last possibility is just your EHE is very slow growing, and so.
Yeah, thank you. Tanya asks, and this is a good question, if all the trials are currently in early stages, how many years out are we possibly looking to find a good drug that works?
Yeah, okay, yeah, very, very good question. So I think it depends. And so it depends on it depends on the individual drug. So how difficult is it to escalate this drug? So if there's a lot of side effects, things can be very slow going as they try to escalate the drug to different dose levels and find the best dose level. Drug is perfectly well tolerated and very little side effects, then that escalation pathway can go forward faster. And if most companies, if they start seeing activity very early on in a particular disease, they're going to try to enrich for that, particularly smaller drug companies. So like, say I have a new drug and um, I don't know, in a rare subtype of lung cancer, there was a response. We're going to start looking for that rare subtype more and more and more and try to enrich that. So the same is true for EHE. So if we start seeing responses in a phase 1 trial of all different cancers in EHE, we're going to try to get more of those patients to prove that. And if we see a big enough effect, then we may not need as large a trial to kind of go to the FDA and say, hey, this is better than anything we got. What do you think? And they may say thumbs up, thumbs down, but it still takes years. I will say that. But if a drug's looking good, and the company's gonna try to make that trial available to as many people as possible to enrich their data to get that drug approved. So if the community's tight enough in EHE that if we found a drug that was better than all the rest, like we're gonna try to get every patient on that trial.
Yeah, yeah. And I think the good news is that along that timeline, along those years that it takes, many people can take part in those studies. So that becomes your good care during that time. Because as you mentioned earlier, you get really closely watched when you're in these studies. So that can be a benefit too. Okay, so I realize we're coming into an hour here. Dan asked another good question. So he says, in the trials, in addition to looking how EHE nodules change in size, do you look at how they change in other ways, such as becoming calcified?
Yeah, so it's a very complicated question, Dan. So if you remember that slide where I said that we were looking at the response for a tumor to make an assessment, is it growing or shrinking? So we're obligated to use this response criteria called RECIST, RECIST. And that's a standard criteria across many different trials. But we know that in EHE and other sarcomas and other diseases, it's a really poor marker of benefit. So we're stuck including that for now, but we're trying to be very thoughtful about including other criteria to show benefit. And our Italian colleagues are looking at things like pleural effusions and how those change. We're very interested in how pain changes. So many of you may know that EHE can sometimes have no radiographic change on imaging, but patient's pain can get dramatically worse. And so we know something's changing, we just can't see it. So we're trying to build all these criteria, but it's going to be slow going. I think we're stuck with this RECIST score for now, but hopefully we can develop these other metrics as we go along.
Yeah, and that's good. Let's see, I just want to make sure that we get through some of these. So Lael asks, any chance that CAR-T or CRISPR could be an option for EHE?
Yeah, so I don't know about CRISPR. So the way CRISPR works, this is a gene editing tool, and the idea here would be to edit out the abnormal gene. It'd be very hard to deliver that to every cell that has EHE in the body. The CAR-T question is interesting. So this is CAR-T are immunotherapies, and the idea here is to you have some marker that's on EHE that's not in other cells in the body, and you can design a T cell, one of those immune cells, to recognize that marker. And so what the way these studies work is they pull out immune cells, grow them in a petri dish, train them to recognize that marker, and then give it back to the patient. And these drugs have been blockbuster drugs for lymphoma, multiple myeloma. They haven't really reached the solid tumor world yet. I don't know of anybody doing this type of work in EHE yet. Maybe Denise does now. She's shaking her head. But if the right marker could be found, it could work. We just have to figure that out.
Yeah. I think there are people that have interest in it, but nothing really far along in hypothesis. Let's see. So Kelly asked, it seems like a lot of patients, whoops, that went away. Hold on, let me get that back. Can live with EHE tumors for a long time without severe side effects. But then when pleural effusion develops, the prognosis gets much worse.
Yes. So this gets at exactly what I was talking about, where patients can have very slow growing disease, can start to grow and then take off. And it's sometimes when we see this pleural effusion show up that things are taking off. And so a lot of us will want to initiate therapy as it's kind of ramping up, but try to start before things like effusions or this accelerated pace of growth starts. It's hard for us to get rid of these pleural effusions once they're there. I've rarely seen that with patients on therapy, but because once it's happened, it's kind of happened. You really need to stop those cells from growing to stop that part. But if we can stop it in its place, perhaps we can make a difference.
Mm-hmm. Yep. Let's see. Fiona asked, would a repurposed drug be easier to get access to than a brand new drug?
This is for running clinical trials. Yeah. So if I'm not sure, there could be two takes on this. So if it's to get access to a drug, what we call off-label. So this is where maybe a drug's approved for a different type of cancer, a different type of sarcoma, different whatever. But we want to use it for EHE. Can we do that? We call off-label. Yes, we can prescribe those off-label. Depends where you live and your insurance. It's much easier to get in the US to get access to IV medications. When we have pill-based medications, it can be a little tricky to get approval. There's different processes depending on what institution you're at. If it's for a clinical trial, then yes and no. So you can get access to the drug sometimes from commercial supply, but you have to fund that study, and it's very challenging in the US to get funding for clinical trials. And so for if I was going to repurpose a drug that was generic, I would need to go to somebody like the National Cancer Institute to get a grant to run that trial, and that's a multi-year process. If it's a drug that's still under patent by a company, I can go to that company and say, hey, I have this idea. Would you fund my study to run this in this rare disease? And they may say yes or no. So there's these different pathways for that.
Yeah, no, that's a good question and something that we'd like to, bring forward, but funding is definitely what we're learning is the hard thing.
Yeah,
to the studies.
It's extremely expensive to run these clinical trials, like even, yeah, so.
We have someone who asked, can EHE change over time and how often do you recommend genetic testing? So that's,
Yeah, also a good question. So we don't know the answer to that is the answer. So something changes, right? Something changes where they're growing slowly and then they grow quickly. And we don't have enough serial biopsy data yet from patients to answer that. But it's a really important question. One that some investigators are trying to look at, including one of my fellows, and because clearly something changes, right? And so how do we figure this out? So we don't know.
Mm-hmm.
How often should you do genetic testing? So for again, it's a hard question to answer. I would say it would be able to think about that when there's been a change in like cancer. So if it was growing slowly and now it's growing quickly, that could be a time to think about it. If a patient was responding to a drug and now they're not, that could be a time to think about it. But these are all a lot. There's a lot of unknowns here, unfortunately.
Mm-hmm.
Genetic testing is quite expensive, so if it's going to be done, you want to make sure it's covered by the insurance company, or some companies will have a maximum out of pocket, things like that.
Mm-hmm. Mm-hmm. Tanya asked, can gene mutations prevent eligibility for a trial? So she has an inherited ATM gene mutation, and would that be an issue? And I think you're going to say probably that's trial specific.
That's unlikely to be impact. In fact, having an ATM mutation might make you eligible for other trials. So I would say that it shouldn't impact eligibility, and might in fact open up some doors because there's a number of drugs looking at ATM mutations. Okay,
good. This goes back to the question that you just answered about changes in the disease. Lael asked, can EHE become aggressive without changing in size much? And she's referencing an article that recommends scanning with PET, waiting, and then scanning again because of the uptake. Thoughts on that?
I'm not following the pet question part, but yeah, so we can see situations where there may not be much imaging change, but a patient's symptoms and functional status are getting worse. And we think that's probably because EHE can be very, we call like infiltrative, or it can be kind of sneaky and grow between tissues rather than like in a big ball like we might see in other types of tumors. So that's why, one, it's important to look at the patient in front of you, not just the scans, and two, why this RECIST scoring is not great for trials, because it doesn't capture that well. But I'll see a patient on a trial or a drug, and the imaging may look exactly the same, but they went from requiring high doses of narcotics to not, and that's really important, right? Like that's a really important lifestyle goal. So these are the kinds of things that we're trying to work with the FDA to address, because like very hard to capture that with our current clinical trial efforts or metrics.
Yeah. And I think that also lends back to making sure that patients are at least consulting with an expert who's understands the disease, who's seen enough of it to understand when you're not seeing changes on scans, but you're seeing changes in the person, as you said in front of you. Yeah. I think that is it. I don't see anything else in the chat or in the Q&A. I think that's all. And we're at about just over an hour. So thank you very, very much. Thanks, everybody. Thank you, Dr. Cote, for your time. Before we close, I do want to put a plug in for one thing. We referenced going to the fightehe.org and looking at the clinical trials page, so you can keep referencing that. We're going to keep updating it at the foundation. We're actively reaching out to companies as they are coming into development with a drug, not necessarily to promote them or to recruit for them or help them. It's really more to find out more about where they're going to have the study placed, so that we can help you as you're looking for places of care and trying to get into studies, just to have some extra information in case your doctor doesn't know. The other thing that I'll say is, if you haven't joined the registry, I would strongly encourage you to consider that. One of the purposes of the registry when we designed it was to help identify participants for clinical trials. So in early phase clinical trials, like most of these are now, that's not really applicable yet, because we're not helping to recruit or find patients for those trials. There's so few slots. But as we get more development, that'll become more important. It does support drug development in a big way. So consider that. Again, you can find that on the website. Stay tuned for upcoming other events. And thank you very much. Thanks, Dr. Cote. This was great.
Right.
Thanks
for everyone. Really great to be here. Yeah.
Thank you. Be well.
Bye.
Bye-bye.